How are you doing? Good? Thank you. Good cry. Good cry. All right. I know nothing about high-dimensional trace. I don't even know how to define this. So I'll focus in this talk on the only word in the title of this workshop that I understand a little bit, which is intelligence. I split the lecture in sort of three parts. The first quarter is me giving my opinion on something. And once I made my point, we'll move on to show a lot of pretty pictures and videos. Is that okay with you? All right. So intelligence, in particular biological intelligence, I'm a biologist. I know very little about math, so I'm going to be lost in a lot of the talks at this workshop, which I love anyway. Biological intelligence is a question that has been studied pretty much ever since modern science has emerged, sometimes in the 1800s, 1700s. You can find examples of people thinking about intelligence as far as Darwin, thinking about biological intelligence. In one of his best book, it's much better. It's actually a book about worms and molds. It's much better than the one you all know about, the origin of species. But in his book, he has an entire section or chapter dedicated to whether or not worms are actually possessing any form of intelligence, which at the time was something that was sort of a controversial idea to express, the idea that intelligence could be something that wasn't just something purely human. Okay? Now, intelligence is one of these concepts. If you look at the history, or if you ask 3 different people in the room how to define it, you'll get 5, maybe 10, maybe 25 different answers for what that word means. And so I spent most of my year in 2023 actually going through a lot of literature to try to figure out what is the best definition of intelligence we have in biology. And found a lot. My favorite one, the one I found the most beautiful, most poetic, maybe the most philosophical, is this one by Jean Piaget, famous child development psychologist, who define intelligence as what you use when you don't know what to do, when neither innateness nor learning has prepared you for the particular situation. Now, it's a beautiful quote. Don't take picture of it. The reason for that is it's actually a terrible definition. Not because he is wrong, but because it actually doesn't match the criteria of measurability for a good scientific definition. So a good scientific definition is something that is universally accepted, that is used by the scientific community in general, but it's something also from which we can measure an outcome so that we can actually do comparative science with it. And if you go back to that definition, you'll notice that Piaget doesn't define intelligence. Actually, he defines what is non-intelligence. And you cannot come up with a measurement with what is not. So I was very sad because I like Piaget in general, and I love that particular quote. And so I dug more and more and more into literature and realized that if I want to actually get a definition that is both measurable and universal, I should look at papers that don't necessarily define intelligence but are testing it. And most of these papers, they'll end up with apparatus like this to test intelligence in different types of animals. So these are 4 of the most famous type of apparatus that are used to test in particular memory, but other sort of intelligent behavior. You have, of course, a variety of these systems. And one thing that they all have in common, right, is that they are trying to see if the animal in this setup it can solve a particular problem, and we give it to it. And actually, in 2019, I recommend reading that review paper by Hamring and collaborator. It seems that essentially the essence of intelligence is problem solving. And I'm not saying that in the sense that it is the definition of intelligence. We are testing intelligence by asking animals to solve problems, like exploring their environment. And so for all intent and purposes, in a practical sense, in a scientific sense, intelligence and problem solving are sort of the same thing. Now, of course, that pushed back the problem to defining what is a solution and what is a problem in the first place. And I'm a biologist. And I come from a tradition of naturalists. And for us, a problem is essentially something that's going to be a limit to the organism fitness. And the solution is going to be a non-random behavior, in this case, that will overcome or push back that limit. I just put that picture here just because I like it. It's nothing specific about the question. But I mean, it is an example of a problem-solving behavior of how do you pass on. For those who don't know, it's how honeybees pass on information about the direction and distance to a particular food source from the famous paper by von Frisch. But anyway, problem and solution, this is my definition of it. And we can also equate fitness in biology to productivity in economics, right? These are the two same things. Fitness is essentially biological productivity. In this particular case, what I call non-random is an ordered behavior, right? Order means that there is a particular sequence of steps that you have to accomplish in order to solve your problem or increase your productivity. You cannot just do things randomly and hope to achieve a particular outcome. Okay, and the good thing with that, for those of you who have done a little bit of, I'm sure Peter is recognizing that very quickly. If you have done any economic theory, any social theory, you know that you can then apply two particular theory to these terms, which is the optimality and rationality, which are very practical in science because they give us bounds on what you can expect the animal to be able to achieve, right? So we looked at the example of search, optimal search strategy, which is essentially what is the best strategy to find X amount of resources or all the resources in the environment. Rationality will be more something that has to do with how you find a maximum amount of the resources given the energy you have available to achieve that goal. But these two things, the fact that we define intelligence as something that increases the productivity of an organism through an ordered set of steps, allow us to reframe intelligence in these two frameworks and give us really nice goals, really nice target that we can compare the animals to. Peter, if I say anything wrong, you just tell me. Now, that question of rationality, optimality, that question of intelligence has been studied most largely ever since Darwin in organisms that possess a brain, right? Not necessarily as complex of a brain as the human brain, but in organisms who essentially are producing behavior through the use of a neural system, okay? And that sort of neurocentric paradigm of intelligence, which is essentially the idea that intelligence is the produce of a network, a relatively fixed topological network of nodes that are exchanging information and weighing and transforming that information. That is essentially what is underlying pretty much all of the current artificial intelligence. Okay, artificial is true, but intelligence in between quotes, revolution that we are seeing today, right? Most of our understanding of intelligence and how it functions is in that neurocentric paradigm. And I'm arguing here, and I'm going to show you examples after, that's where the pretty pictures are coming in, that intelligence in biology, especially if we define it in that problem-solving framework, can take many different forms. And the two examples that I want to highlight, one of them you probably are more familiar with already, or at least some of you are, which is the paradigm of swarm intelligence, which is a paradigm where the main difference with the neurocentric paradigm is that the connection between the units are largely random, or at least they are extremely fluid. You'll see some people actually referring to this type of intelligence as liquid intelligence, which we can argue whether or not it's a good analogy with physics. I prefer gas in that case. Anyway, I have to have a discussion with Ricard Solé on that. But this is the type of intelligence that we'll find largely in all large social groups on the planet, ants, flocks of birds, schools of fish, etc. And then the last kind of intelligence that I think is completely understudied, even less than swarm intelligence, is the question of what we call basal cognition. I don't know how to represent it, but with a blob, and you'll see that later. And the question of basal cognition essentially is what happens now if you don't even have organisms, no little particles like this that have all their own little neural networks and interacting with each other. What happens when you remove completely the concept of neurons from the equation, right? Can you be intelligent? Can you generate intelligent solution to a problem without neurons? And this is something that you'll find, tree, bacteria, politicians. It's my political joke of the day. And what I want to focus on this talk is I'm going to completely ignore neurocentric paradigm. Enough people are talking about it already. And I want to focus on one of these two stories, or actually both if we have time. And so that's the point where you can choose which one you want to start with. Do you want to hear about ants and the cool stuff they can do, or do you want to go talk about yellow goo?
Basal cognition.
Goo.
Yellow goo.
Who's for goo? I don't need to ask for the ants because apparently there is a majority for the goo. So let me just jump to that part. Funny, most people prefer to start with the ants for some reason.
Most people start with dog and cat too. So. Not this group.
I appreciate the non-determinism.
Hmm?
I appreciate the non-determinism.
Okay. All right, so basal cognition, right? Basal cognition, or basal intelligence, however you want to call it, it's a sort of an emerging field at the moment. It's one of this field where half of what's published is nonsense woo-woo, and half of what's published is actual really hardcore science that is making progress. I'm hoping by the end of today, I'm on the hardcore science part of it, but you still find a lot of interesting woo-woo there. And essentially, basal cognition is trying to understand how organisms. I need to do that here. How organisms that don't have brains, don't have neurons, are still capable of solving problems. I mean, we're talking about search strategy. A tree has a huge root system, sometimes much larger than the emergent part of the the above ground part of the tree. Sorry for the English is not my first language, as you know. So sometimes I make mistakes like that. But that network of roots, essentially, its job is to search for resources. And I'm sure Peter can tell us more about this. But if you look at the pattern that these roots are adopting, they look, if you analyze them, as some sort of pseudo-random walk. And so there is some form of problem solving happening here on how to exploit the resources that sometimes can be very far from the trunk of the tree. Other organisms, of course, are unicellular organisms like bacteria, yeast, etc. And the reason why it's so important to study intelligence in these systems or problem solving in these systems, so I don't have a problem with cognitive scientists, is that essentially there are a lot more of them than there are of us, right? This is just an estimation from an old study, 2018 study of the biomass of all that's living on the planet. And essentially, everything like that little gray box, it's all the animals. This is everything that has a brain. And everything else is everything that doesn't have a brain. Right? So organisms have survived on this planet long before neurons have appeared. They have solved problems long before neurons have appeared. They also are just by sheer biomass dominating the planet. And yet, the way they solve this problem is largely understudied. And I'm hoping to encourage some of you to jump into that bandwagon. So come with me and do serious science about these questions. So it's very important, and it's a question that I like to reframe in a more like a humoristic tone, which is, this is what we call the Homer-Simpson paradox in my lab. The Homer-Simpson paradox is essentially the problem of how do you become successful without a brain. So you all know Homer Simpson here, right? Homer Simpson is probably one of the least intelligent person on this planet. We all agree on that. At least he is portrayed like that. But he is also one of the most successful person I've ever met. I mean, he's the director of security at a power plant, at a nuclear power plant, and the entire history of the power plant, no major catastrophe has happened under his leadership. He's been to space. Who else here has been to space? Yeah, no one, right? You have not been to space. He's been to space. He's met pretty much every president alive, and even some dead presidents. Right? He almost became world champion in boxing. I don't know if you've seen that episode. It's a sad episode where he ends up losing, but for the right reason. But anyway, by any measure, he is a lot more successful than you and me, right? And yet, he doesn't have what we typically call the brain. And so in my lab, a long time ago, a postdoc of mine joined the lab and told me, can we study the Homer-Simpson paradox, please? And I was like, how much is it going to cost? And he said, not much. He said, okay, you can do it. And so we started a program on studying that question in organisms that needed to have two particular characteristics. It needed to have no brains, no neurons, and it needed to be yellow, like Homer Simpson. And so we chose to work on that organism here, which is the Slimol-Phasarum polycephalum. Some of you probably have already heard of it. This is a unicellular organism, so all that yellow mass that you're seeing is one single cell, which is kind of impressive. It's a syncytium, meaning that it's one single cell, but it's millions and millions of nuclei that are floating inside the cytoplasm of that cell. And it's an organism that has been originally largely studied for the property of its cytoskeleton. So there's a lot of studies in the 1960s, 70s on how the cellular machinery inside the cell was working and was sort of abandoned in the 80s and 90s, and sometime toward the end of the 90s, starting to make a comeback when people realized it has actually really cool behaviors as well. And some of these cool behaviors are things like the ability to solve a problem that is relatively complex, which is the problem of solving what we call the U-trap problem. So it's a problem in cognitive science. If you have an organism and there is a gradient, so there is a directionality in the gradient toward the food source, and you place a barrier, a U-shaped barrier in the middle of the path, an organism that is not capable of spatial memory typically won't be able to recognize that he's already visited that part and won't be able to escape the U-shaped trap. It turns out the slime mold is capable of doing that, not because it has an internal memory of the space, but because it marks its environment with a chemical that essentially create a trace in the environment of where it's been, and that later the slime mold is going to avoid. So it has an external spatial memory, which is a very interesting concept in general. So it's capable of solving problems like this that several, highly intelligent birds are not capable of solving. It's also capable of optimizing transport networks. So this is a very famous experiment from Nakagaki's lab in Japan, where what you see here is a piece of slime mold. Every single of these little dot is an oat flake that essentially is really good food source for this organism. And they are placed here in this particular pattern, which represent all the major train stations in the Tokyo area. And in that experiment, what they have done is essentially look at how the slime mold connect all these different resources with each other and demonstrate that the property of that communication or transportation network are not that far from, no, I don't want to say optimal because I don't like that word, but very, very efficient, even compared to what the engineers that have developed the Tokyo rail network have come up with. It's not a match one-to-one, but it's a very efficient system. And all of this is done all this optimization is done without a brain, without a single neuron. They're also capable of solving fun problems like this, again, from Nakagaki's lab in the early 2000s, which is essentially finding the shortest path inside a maze, which is a problem that is, even for young children, relatively complex to solve. And yet the slime mold is capable of finding a solution in that problem, and often the shortest path is actually found. Fun story for these two studies, Nakagaki received twice the Ig Nobel Prize. And I think he's probably the only person I know that has received it twice. For those of you who don't know the Ig Nobel Prize, it's a science that makes you laugh and then make you think. Right? But essentially, all what this is showing is that when faced with problem of optimization, problem of searching, etc., the slime mold, for example, polycephalum is actually fairly good at solving this problem, despite not having an internal cognitive machinery, at least not a classical one, as we usually think when we talk about intelligence and cognition. And so that question, I started, so Chris Reed is the postdoc that started it all. Chris now is back in Australia at Macquarie University. And these are the list of all the people that were involved one way or another in the project. In red, people that were in my lab at some point. In black, external collaborator. And we started trying to understand that question in using a paradigm which, Peter, again, I'm sure you're very familiar with, which is the multi-arm bandit problem. So you know what the arm bandit is? At the casino, it's the slot machines. A slot machine is a system that has a certain payout. Each machine has a slightly different payout at the casino. And if you want to find the machine that has the best payout, you have to try all of them. And every time you have to pay some money. So you have an exploration phase that costs you, and that allows you to gather information about the environment until you decide which is the best machine, at which point you switch to an exploitation phase in which you only focus on that single machine to try to maximize your payout. Even if you find that machine, you will lose. The casino always wins. But that's not the problem. It's a really good paradigm to study that problem of how do you switch from exploration to exploitation, and whether or not that switch allows you to consistently find the best solution in the environment. So the experiment is quite simple. You have a piece of slime mold that starts in the middle of that piece of agar. On each side, on the left and right side, there's a number of darker little pieces of agar. These are actually contain something nutritious, so something the slime will like. And you can see that at the beginning of the experiment, the cells start expanding sort of equally in both directions. This is the exploration phase. And after a phase of exploration, it ends up stopping its growth in one direction and only pushes in the other direction. I know I'm going to get that question. I said growth, and it's incorrect. The biomass of the slime mold barely changes during that experiment. Right? The timeline of that experiment is short enough that the biomass doesn't significantly increase. Right? So what you're seeing is essentially the slime mold spreading its biomass over the available surface. And then you can design multiple of these machines which different payouts, and you can actually increase the complexity of the system from extremely simple cases where most of the food is on one side and no food is on the other side, all the way to cases where the amount of rewards and the number of reward sites is random, but also how much, I don't know if you can see that tiny little number in the middle, but how much you get as a reward every time you hit one of the sites is random as well. So this is probably the most complicated case of solving, is that you don't know how often you're going to win, and you don't know when you win how much you're going to win. The only constant in all these experiments is that one side has always more nutritious value than the other side. Okay? And if you test your cell inside all these different scenarios, what you see is that consistently the cell choose the correct arm, the arm that has the highest nutritional value, in at least 75 to 80% of the cases. Okay? And just for reference, this is about as good as what pigeons are going to do in a similar setup, less good than what humans are doing. But this is a pretty impressive feat if you think that what you're looking at is essentially a big sack of protein and fat and water. Right? It's nothing more than that. Now, when we saw that result, we immediately wanted to know what happened. Of course, how do you explain that the slime ball can solve that problem? And especially, how do you explain that it can solve all these different classes of problems, especially what we call here the non-binary problem, which is a very complex problem where you can't predict ahead of time how often you're going to be rewarded and how much the reward is. And so typically, you have two hypotheses in this case. The slime mold either has, for each of these class of problem, a specific solution that it implements, and a different mechanism. And this is something like Peter showed earlier. For such strategies, you can have different classes of such strategies, and some organism can switch from one strategy to the other as a function of the environment, stage of life, etc. Or you can have one single mechanism, the ring, that bring them all together, and it's capable of solving all these different classes of problem. In other words, there is only one, if you want, mathematical equation that can, depending on the context, lead you to different, not necessarily optimal, but efficient solutions. So you can imagine which one is going to win, but let's pretend you don't know which one is going to win in the end. So to study the slime mold, there's something we need to understand, is how that thing moves. It essentially moves by spreading this sort of like fingers. Each of these fingers becomes essentially a tubule of a tubular network. And inside that network, you have cytoplasm that is flowing and connecting the different parts of the cell to each other. Right? So you can think we can simplify, if you prefer, this very complicated network to essentially a single of these tubules. We have developed a method where we can grow the slime mold on water surface. And when you grow it on water surface, it's either making a really nice linear tubules like this with very few branching that we can excise. That's one of the other properties of the slime mold. If you cut a slime mold cell in half, you have two slime molds, and then you put them next to each other, they fuse, and then you have one slime mold. It's a magic trick in biology. It's really also practical because if you need to produce more and more slime mold, you have to do is cut cells, put them on a petri dish, and then the day after you have a huge incubator full of slime mold. So if you want to do research on slime mold, super cheap. You need oat flakes, water, agar, and a reasonably skilled graduate or undergraduate student. That's all you need. So we can cut out your piece of slime mold into a single tubule. which has the advantage now that we can study that single piece of the slime mold. We can put it in between two food sources, one that is a high-quality food source and one that is a low-quality food source. You can read the papers about the exact composition of the food sources. And then we can look at the different section of the tubules, and we can measure how that tubule essentially change shape locally. So I think I should have shown this in the previous video. But if you look at the tubules, you can see there is some sort of like oscillatory behavior where they expand, contract, expand, contract, expand, contract. And so that's what we do here, essentially. We are measuring in each of these different cells that contraction expansion activity. Okay? And once you have that, you can essentially try to use a method. It's called transfer entropy. Everybody knows transfer entropy? Okay. Essentially, it's a method that, to simplify, try to tell you if I know something about what's happening here, how much does it help me predict what's going to happen here, and vice versa. Okay? So it's a method to determine who essentially inside that tubule is controlling the behavior of whom. Is there a part of the slime mold that controls the behavior of anyone else? Now, I'm a typical animal behavior person, and in animal behavior, the typical baseline expectation is that wherever there is the highest quality signal, that's where you're going to have the highest amount of information that's going to be sent out from there to recruit individuals toward the high quality food source. Think of ants, they find a good food source, they're going to recruit using pheromone, and the higher quality the food source is, the more recruitment there's going to be. If I find a candy store around the corner that's super good, I am going to advertise this like crazy. If you find a, broccoli store on the other side that nobody likes, you're not going to talk about it very much, okay? And so you expect normally that the part of the cell that is in contact with the food source is the one that's going to emit the most information, control mostly the behavior of the slime mold. Turns out, no, slime mold is weird. It does the complete opposite. The part of the slime mold that control where the biomass is going to move is actually the part that is in contact with the lower quality food source. Right? So it's a form of leading from behind, if you want, for those of you who have done some collective behavior. It's not a leading from the front. And that is a bit of a strange result, at least not the expected result. The information seems to be coming from the side that is actually very far from where you want to be is.
I'm not a biologist.
Oh,
Like
I pretend to be a biologist as well.
I'm like a baby chimpanzee, actually. So this was very intuitive to me because as a human, you go to like Yelp, you're looking for bad reviews because most likely
Are you looking for bad reviews in Yelp?
Yeah, because most likely people are going to bad review than good reviews. I actually find that I'm more likely to bad review a place when it's bad than good review. So for me, this was very intuitive. I think it's telling you, don't come here. It's bad here. Come, when it's good, it's like, I feel bad for my dentist. He's really good, and I haven't put a good review because if I put a good review, more people will go, and then I won't be able to get an appointment. That's what I thought would be happening here.
We should have a discussion about game theory now. Okay. Yeah. Oh, there's no question. Oh, by the way, we can question anytime you want. If we never finish that presentation, it's fine. I gave a lecture at a summer school a few weeks ago. We didn't go past the definition of the first 3 words on the title slide. So.
This is amazing. So
if you think this is amazing, wait to see the rest after.
I've read some of the stuff, so I'm very curious and excited to see the rest. So if the food source is oscillating between the two sides, it is helpful to have some information coming back from the low side in case things change. And in some search algorithms, also in reinforcement learning, there's some incentive to go to places you haven't been in a while. Is that the kind of thing that's happening here?
No. So I'm going to argue actually for a case where no information is passed.
From even the low
for
side
even
to
side.
From either side.
Right. The mechanism I'm going to show in a minute is a mechanism that actually requires no communication whatsoever.
How long does the oscillation or communication take if it happens?
So this oscillation, this pumping behavior that you see, this like expansion, contraction, actually I have a video after I can show you that's how it looks like inside the cell. This is actually live, right? Like that's not sped up or anything like this. If you have a microscope and a slime mold, you can see the cytoplasm flow in real time. It's the fastest cytoplasmic streaming that we know of in the living world. That movement of the cytoplasm back and forth, it's has a frequency of 1 to 1.5 minutes, depends on conditions and state of the slime mold and size of the pipe and etc. But it's about between a minute and a half typically. Yes?
Sorry if you mentioned this already, but what is dictating the motion of where the cytoplasm goes?
Well, we're going to come to that in a second. I'll explain the more like the physical part of it. Any other question? All right, then I'll continue. So that mechanism, that flow of cytoplasm, essentially it's the results of the presence of actomyosin. So actin-myosin is a protein complex that lines up the membrane of the cell. These are the same proteins that are actually used by your muscles to generate force, right? So these are proteins that can polymerize or depolymerize and generate contraction or relaxation of the membrane, the same way your muscle fibers are generating contraction and relaxation. The chemistry is a bit different between us and them, in particular the relationship to calcium, but this is not something you care about, so I will pass that part. But what's important here is we have to remember about this calcium is that essentially the cell contains reserves of calciums inside, and when the calcium is released locally in the cells, that will actually break down that polymer. And the result of that is that locally in that area, there is less ability to maintain the structure of the cell, and the cell is going to expand locally. On the contrary, where the calcium is being recaptured inside these vesicles, the polymers are going to be able to form and will be able to apply a contractive force on the membrane. And then there is an entire internal cycle where essentially when calcium is released inside the cytoplasm locally, this vesicle will start working to recapture that calcium. And so you will end up with that cycle of a sort of peristaltic cycle of contraction relaxation, cyclical cycle, which is essentially the result of that capture-recapture of calcium by those vesicles. Does that make sense? What's important is that some part of the cell will contract, some part of the cell will expand, and this is controlled through a calcium release, calcium diffusion, and then calcium recapture. All right, that's nice. Now, what happens when food is present in the environment? There are sensors on the surface of the cell, and these sensors are essentially proteins that are connected through a chemical cascade to these vesicles inside. And what they do essentially is locally I sense food, I trigger the release of calcium. If I trigger the release of calcium, I trigger the dismantlement of that muscle, if you want, lining, and locally my cell is going to expand, right? And what that creates essentially, if you've done a bit of physics, if this part of the cell now has become a sort of flabby balloon, and this part here is like a strong contraction, strong pressure, that creates a natural gradient of pressure that moves cytoplasm from that part of the cell to that part of the cell. And this is done without information from the food being sensed here ever traveling back here. This is just done here by the relaxation of the membrane locally. Does that make sense? So there's like similar things in collective behavior. You may have seen this paper on fish school capable of following a gradient of light, or more exactly a gradient of darkness. And this is done by the fish that are in the dark, slowing down, anchoring the group where the darkness is. And if you move the darkness spot, these fish now are in the light, they start accelerating. The fish now that are in the dark slow down and anchor the patch somewhere else. So it's a mechanism essentially that is not an active mechanism of movement. The movement is created by passive, by this part of the cycle becoming more passive. Does that make sense? Now, it's math thing, so I put a bunch of equations. I don't know what they are. I didn't come up with them. But these are essentially from an old paper by Teplov that has been reshuffled by a former PhD of mine, Habib, to essentially model that system as a field, a 2D field, where we can essentially simulate the cell thickness as the third dimension of that field, if you want. So locally, what we're simulating in that here is how thick the cell is at one location, which is an indication of how much biomass is at a particular location. Now, with that model, we can start looking at whether that model reproduces some of the typical behavior that we see of the cell. So this, if you look at the calcium inside the cell, you will find that the calcium inside the cell creates waves of calcium that are traveling through the cells. So these are these waves of calcium that are creating that cyclical pattern of activation of a contraction relaxation. And then you can see that we can generate these waves with the model spontaneously without having to do anything, but that's not the interesting part. Now, what happens when we place them in a gradient? Can they climb that gradient as well? Can the cell climb the gradient? And you can see that the cell is capable. So if we place a grain of food in one direction, the cell is going to naturally migrate toward that gradient because on that part of the cell, it becomes more relaxed. That part keeps contracting at its usual rhythms. There is a natural gradient of pressure, and the cell biomass then naturally migrate toward the food. Can we do some network optimization? Of course, we can do some network optimization. Not as complex as this because the simulation takes some time. So we did a much simpler version of it with only 3 food sources. And you can see that the cell starts in that typical pattern when it expands sort of equally in all directions. That's one of the beauty of the slime mold. It doesn't need to do a random search. It can search systematically in all directions until it finds something good enough, and then start simplifying its structure to reduce essentially the biomass that is used for further exploration and maximize the biomass that is located at the food sources. This is like further, I say this simulation can take for very long, but you can see that after a while, you have a nice minimum spanning tree that has been created from these simple properties. Now, can we recover that behavior? Remember that behavior where we know that we find that the low quality side is the one controlling the behavior. Now you know how it works, so you should understand why that will be the case. In silico, we recover that pattern of influences and becomes normal to explain now, right? The side that keeps pumping is the one that creates all the work in the cell. So naturally is the one that's going to control how the other side of the cell that is not doing any work anymore is behaving. And finally, can we win at the casino? And that's my favorite part. We did the experiment, so this is the simulation of the experiments, and you can see that the simulated cell does the same sort of systematic exploration in both directions and then stops moving into one direction and then concentrates its movement toward the other direction. And actually, when we simulate the same scenario that I've shown you before in silico, you find very similar pattern of results. And you can find as well that the places like these blue cases, which are where randomness is introduced, which is harder to solve, are also harder to solve by the simulated cell. The place where there is more stability in the distribution of the resources, which are these green bars, are easier to solve. And the one that is supposed to be the hardest to solve is also the hardest to solve in silico. So we recover a lot of these different behaviors that are all coming essentially from a single mechanism, a single optimization mechanism that can be applied to all these different situations. Because if you think about it, all these different situations are a problem of how do I move my biomass from somewhere I don't want to be towards somewhere I want to be. So it's a search problem, I guess. But this is a strategy that you probably have never encountered before. Actually, I would like to talk to you about something after that, because I have another search strategy that we just discovered that I don't think exists anywhere else in the world. So I'll see what you think about it after. And so this is just to summarize basal cognition, right? This is an example of basal cognition. And typically, most examples that we know of basal cognition, they have three properties. One is that they are achieved without the need of a neural network, so you can generate intelligence without a brain. They're often physics-based, right? They essentially is the organism exploiting its own physics. Instead of coming up with computation or mechanism to compute and then make this computation trigger then mechanical actions, here both the computation and the mechanisms are the same process. which is, I think, something also that is understudied in general. And they are often multi-purpose. The same physical property can be then reused for other actions. And that's going to be it for the basal cognition. Do we have any question, or should we still do the ends? How much time do I have?
You have 19 minutes.
Oh, we can do the ends. Do you want to do the ends?
Yes.
The ends. The ends. I have to go back. All right. It's not ends. These are not ends. So we looked at basal cognition, so this idea that we can generate intelligence without even neurons. I'm going to backtrack a little bit, and we're going to come back to organisms that have neurons, but where the computation is not done directly in the neural network itself. The computation is done at a level above the neurons, and this is typically a field that we call swarm intelligence, which is a sort of sub-branch of a larger field called collective intelligence. So for those who don't know what collective intelligence is, if you remember my definition of intelligence, if we have two individuals, we pose a problem to them, each of them has a certain output, and we sum these outputs, they'll give you a value, which is essentially the sum of the intelligence of all these organisms. Now we have them work together, and by working together, there's some form of synergy in their work that is created by the interaction between them, and that leads to a larger outcome, either for, on average, each of the individual, or for the individual that had the lower outcome before. Okay, I'm not going to delve into all the reasons why collective intelligence can work. I'm going to focus specifically on swarm intelligence. The swarm intelligence is how you achieve that through a particular mechanism, which you're probably all familiar with, self-organization. Who has never heard of self-organization here? Very good. I'm going to still talk about it and define it because I have a really great video to show you. And I will be, I'm always sad if I can't show that video. But self-organization, just as a reminder, is a system that starts in a disorder state of agents that are coming together and through some mysterious force start interacting with each other, and the result of that interaction is a new behavior, a new organized behavior. What's really important in that behavior, it's the cutest video you've ever seen, right? You can type Scotty Pinwheel on YouTube and you'll find that. And then you'll find there's a lot of other people that have tried to replicate this with other breeds of dogs, with cats, and all sort of pets and animals. It works every time. But what's important here is that you can see that at the end, when the original source of the attraction, when the objects are not forced to interact with each other anymore, the organization falls apart. So it's a form of dynamical organization, right? They're so cute. You want to watch it? You want to watch it again, or? Yeah.
Oh my goodness. Watch it whenever we need it. If you could send it out on Slack so we can watch it.
I don't have you. I'm not on your Slack, but I'm sure someone can do this.
Okay.
Oh, it's my first time here. I don't know any of how this program works. I don't even know why I was invited. The person who invited me is not even here. So if Dora is listening, she knows I'm talking about her. But what's important here, right? It's a form of organization that is a dynamical form of organization, and that requires constant input, constant energy input in order to keep functioning. Now, swarm intelligence and self-organization are not the same thing. Self-organization is a principle on which swarm intelligence can be built, but you need more than self-organization to generate an intelligent collective behavior, right? Remember, we have to have a problem to solve, right? In the case of the little Scotty Pinwheel, there was no problem to solve in particular. If anything, that collective organization was getting into the way of them accessing their food. And that's where something like natural selection, when you combine it with self-organization, is a mechanism that's going to be able to tune that self-organization in order to generate collective behavior. Now, if you're an engineer, you replace natural selection by design or by any form of optimization algorithm that you like. But self-organization by itself doesn't necessarily generate intelligence. It needs to be combined with a mechanism of optimization, right? And I will argue that these two mechanisms together, these two theories combined, explain most of what we understand today of how life emerges and functions on this planet, right? Even people back in the 90s argue that natural selection was just a byproduct of self-organization. I'm not going to go as far as that. I think they are two distinct mechanisms because they act on two distinct levels of the system. But you need to combine these to be able to solve problems. And I'm going to give you an example of how self-organization combined with natural selection has led to the ability of a particular species of ants to solve a very difficult problem, in my opinion. So what you see here are ants. You've all seen ants before? Have you all seen ants do that before? All right, so if you want to see ants do this sort of constructions, which are built out of other ants, essentially these are ants all attached to each other. Everybody is alive in that structure. And this is essentially a temporary nest that is built by army ants out of their own bodies, right? Everybody is alive. We sometimes call these sort of structure living architectures because it's literally an architecture that is constantly reshaping itself, constantly living. And the reason why these ants have to build this structure and have come up with that solution, wrong button. You're right, it's not an intuitive thing. Is that, so these ants are called army ants. They're not a taxonomically delimited group. For the biologists among you, you understand what that means. For those of not biologists, you don't have to worry about that. They are very weird among the ants. Among the ants, they don't do like your typical garden variety of ants behavior. In particular, they are a top predator in their ecosystem. Nothing eats them. Actually, they have a lot of birds that are parasites of them, like any large carnivorous has parasites. These ants have parasites. Probably the only example I know of an organism that has parasites that are bigger than themselves. And one of the characteristics of these ants is they are completely nomadic. Part of the reason for that nomadism is that they have strict reproductive cycle that force them, and they are carnivorous, and that force them to regularly change location because, being a carnivorous organism means that you're eating what's at the top of the food chain. And that means it's a thing that depletes the faster, so you need to move and go to other locations more and more, very often, to be able to find new resources. But they are really nomadic. And so unlike other ants that are sedentary, they do not build nests. So these are examples of nests. If you have never read that book, I highly recommend you find it by Walter Chinkel, Ant Architecture. It's like a history of the structure of underground nests in ants. But army ants do not do that. And because they are nomadic, there's no point in digging into the ground, building a very complicated structure on the ground to abandon it about every 2 weeks, they just migrate to a new location. And then during that migratory phase, they change location every day. Okay? And instead, when they are migrating, so this is if you go to the tropical forest in Panama, in Central America, South America, you might stumble upon one of these raids, or one of these swarms of army ants, and you can see they have to migrate over terrain that are extremely, extremely chaotic, extremely unpredictable, that have structures that are extremely different from one place to another, and yet they have to be able to pass all these terrains in order to move on to their next hunting grounds. And in order to optimize that movement over this very complicated terrain, they've developed that physical and behavioral adaptation, allowing them to essentially attach to each other in ways that are extremely robust. That's the advantage of an exoskeleton. You can let all the forces rely on the exoskeleton, and you can stay in that position without exerting any force with your muscles. But they are capable of attaching to each other to essentially form a structure that can be extremely large, as I showed you in that first image. So this is a much bigger project. A project I started when I was a postdoc with Ian Cousin at the time at Princeton. One day Ian dumped on my desk a bunch of old data that he never finished to analyze, and he said, I think he was worried that I would never publish anything, so he just dumped all of that on my desk and said, can you finish that project? And that was in 2010, and we are 14 years later, and I'm still working on it. I published on it in the meantime. But these are all the people, and as earlier, in black are external collaborators, in red are people that work directly into my lab on that question. I guess I'm not going to tell the story in order, but I'm going to tell it in order of the logic of the mechanism. One of the most recent experiments we've done, actually, is an experiment where we challenge the ants with a structure, which is a bridge. Oh, I should mention, all of what you're going to see are research that is done in the field, right? So I don't know if any of you does research in the lab, it's hard enough. Everything I'm going to show you is done in the field. So it's, in my opinion, another like whole set of problems. But you're on a tropical island for 6 months. It's not bad. But then experiments, right? We force the ants to walk on the platform. That platform has a movable platform in the middle that we can tilt at different angles. So we can keep it flat, which is the easiest ground to walk on, and we can tilt it up to 90 degrees, which is essentially vertical. And what we noticed, if you look at, on this platform, if you look at the proportion of the ants that are slipping or falling off of the platform, in green here in the first 5 minutes of the experiment, or in yellow in the last 2 minutes of the experiment, so in the first 2 minutes of the experiment to the last 2 minutes of the experiment, you can see that as I increase the tilt of the platform, so it becomes more and more vertical, the number of ants falling off the platform increases. But after about 10 minutes, so the last 2 minutes of the experiment, that number has drastically reduced. And the reason for that is that, and you see in these 2 videos, is that as time passes, so this is the vertical, completely vertical surface, this is 50 degrees, so about this incline, it's already pretty steep. The ants actually are going to start anchoring, some ants are going to start anchoring themselves into the substrate of the platform, forming a form of scaffolding that actually help the rest of the colony pass across that difficult terrain, right? And you can see actually, if you look at just the first 5 minutes of the experiment, as you can see there's a nice correlation between the number of ants that are falling off of the platform and actually the size of the scaffolding at the end of the experiment. So the harder the terrain is to walk over, the larger is going to be the scaffolding that you build. And that, if you've done a bit of cybernetics in your studies, can be explained with a simple thermostatic loop, if you want, where an ant that is going to cross a particular surface, estimate the difficulty of moving on that surface, and the most difficult it is to walk on a particular surface, the higher the contrast, the higher the probability to join in the structure. By joining the structure, they make it easier for the next ants to actually walk on the structure, so they diminish that constraints, which reduce the probability of the next ants to join in the structure. Right? It's a simple thermostatic loop, and you can simulate that, right, and with the correct parameters adapted to the experiments, and you can see that that simple thermostatic loop explains very well in different degrees of difficulty how the size of the scaffolding is growing. And even in simulation, if you introduce that probability of falling inside the structure, you can see that the simulation, which are the blue bars now, in the first 2 minutes of the experiments, as before the simulation, more ants are falling off of the structure. After the scaffold is built, less ants are falling off the structure, both in the experiments and the simulation. So a simple thermostatic loop is enough to explain that construction behavior. I build not because someone tells me to build, I build because it is hard to walk on that surface. All right. Now, how do you decide not to leave the structure? Because the structures are not permanent. They are going to disappear spontaneously after a while. And we did a second experiment. So that was the first experiment. So we're going back 10 years in the past. Simple experiments. We didn't have any fancy stuff to bring to the tropical forest. We find a bridge. We take tweezers. We destroy the structure. And then we see how fast it rebuilds. And you can see here that the traffic on the bridge recovers in about 30 seconds after destruction of the bridge. Okay, so extremely fast recovery from a 100% damage to the structure. Now, what we measure then is we look at the ants joining the structure and we measure the individual probability to join the structure as a function of what we call here the packing fraction. So if you prefer, the packing fraction is how already packed the bridge is, how many ants there is already inside the bridge. And as before, right, if the bridge is completely formed, the probability that you join the bridge becomes very low. If there is a high difficulty of walking on the bridge because there's a gap is open, then you have a high probability to build or participate in the structure. So that's the same thermostatic loop as before. So we have 2 experiments confirming the same mechanism. Now we look at the time the ants spend inside the bridge. And we have recording up to 500 seconds because well, the undergrads didn't want to look longer than 10 minutes at the experiments. But you can see this is a really nice exponential fit. And actually, we have evidence from the field that some of the ants might be stuck in this structure for as long as the structure exists, right? So it's not a behavior where they get fatigued or anything like that. And we look at this as a function of the flow of ants over a given ant. So an ant is in a bridge attached, and it's essentially sensing other ants passing on top of it. They have this little hair everywhere that sense touch. And you can see that if an ant senses a very high traffic rate over it, it stays very long inside the structure. If the traffic rate diminishes, it immediately leaves, right? And that, essentially, you can add that back to our little cybernetic model, where if I am in the structure and I want to decide to leave, if there is a lot of traffic, that will inhibit my probability of leaving, and I'm going to stay inside the structure. If the traffic dies down, I will immediately leave the structure and rejoin the colony. Yes?
You're also not a biologist. Ants have brains, right?
They have brains, yes.
The brains are involved in this.
Yeah, I didn't say they were not. I said we were backtracking from the basal cognition.
I know.
Yeah, they do have brains. They have tiny brains, though. I mean, there are more neurons in your pinky finger than there are in an ant. But essentially what we have here is a mechanism by which the constraint of movement decide if you stay, if you join, and whether you are useful decide if you stay. And we can show through simulation that these two simple feedback loops will produce exactly the behavior that we observe in the field. And that has also some interesting consequences. I promise this is the last bit of it, but this is the coolest part. Because there's something we always wanted to explain, and we could not explain until we figured out that full double loop, is if you go in a tropical forest, you often find bridges, this structure that they build across these gaps that are suspended, right, and much longer than the size of two ants. So there's no way they could have been done by reaching over to another one, right? Now what we found is we hypothesized that the bridge wasn't built there. It was built somewhere else and then transported. And transported being a big word here. You'll see it's not really a transport in the classical sense. So we designed an experiment in which, again, we are looking from the top down in a particular case, right? So the ants are in a completely horizontal surface. But we force the ants to go through a deviation along the trail, right? So we add a cost to their movement. And you can see already that at the top, the ants, if they can walk over the gap, they will walk over the gap. Of course, walking over the gap is a bit more difficult than walking over that plastic surface, and ants will tend to start building at that location. Now, ants, as you may know or may not know, they like to walk the shortest possible way. So, and like we do, so if they can shortcut somewhere, they will shortcut. And meaning that at that particular location, there will be more traffic than here on average. If you remember, more traffic means I stay here longer, less traffic here means I leave. And as a result of that, if you let the game play, this takes about 30 minutes in real life. The ants dismantle, the ants that were attached here leave, and new ants are attached at the bottom, and that suspended structure start moving down the structure, down, toward the opening of the gap. And essentially, every time they do this, they shortcut, right? They save all that walking time. And actually, we can do some statistics on that, right? So that's, if you look at the distance that structure moves down that deviation, it's moved fast at the beginning and then start slowing down. And more importantly, it depends, how far you move depends on the angle between the two branches here. So if you have a very narrow angle, meaning the deviation is actually you have a lot of deviation to take into account, the bridge is going to move much lower down that structure and so shortcut even more. When the deviation is more flat like this, if you want, so there's less change in the movement of the ant, that is going to move less far down and shortcut a lot less. And interestingly, right, we can reframe all of this into a cost-benefit framework, right? With the benefit being how much you save and the cost, how many ants you have to put in there to build that structure. And of course, if you were to move the bridge here as low as it was here, you will need a lot more ants. So the cost will increase actually if you look at the cost-benefit curve. Right, the cost increase much faster when you have large angles or low impact deviation than when you have a deviation. And then working with some mathematician, we actually reproduce that cost-benefit curve in a mathematical model that you can see fit nicely what the experimental data are giving us, suggesting essentially that that simple two-feedback loop, it appears to be simple, but it has like really high level of consequences in how the whole colony can optimize overall its environment, its movement through the tropical forest in this very chaotic environment. And before, because the question is going to come, but this is just a tiny little deviation. They can build 100 of these bridges along a trail, right? So every time, if you compound this over the whole colony and the whole like exploration or migration trail, we've done a back of the envelope calculation in the paper. It's in the supplementary material of the paper. It can be up to 20% of the ants are trapped in these bridges, right? So it is a significant cost for the colony, and so it better bring in significant benefits in return. And I'm just finishing, and then I'll come to you. This way I'm done with the talk, and you can kick me out. But this is swarm intelligence, which is a different paradigm of intelligence, right? Instead of having units that are connected through a topologically fixed network, we have disconnected units, so the network of interaction is constantly changing. It's fluid or liquid, if you want. It is done through purely local sensing, right? The ants only sense how hard it is to walk where I am right now. It's only sensing how much traffic there is on me right now. It doesn't need to have any form of long distance sensing. And it can be achieved here, again, without explicit communication, right? In this particular case, no two ants is talking to each other. They only gather information through cues left by, oh, I'm being walked over. That's a cue that there is a lot of traffic. But you don't need to evolve complicated or explicit communication mechanism to be achieved this form of intelligence. And with this, there was a question.
Thanks. So I was wondering if you repeat the experiment with the same colony, is there any form of memory, or do they learn the same thing?
So I wish I could answer that question. This is, again, field work. And it is really, really, really hard. You don't know that you're going to find the same colony at the same place the day after, because they move constantly. Every night they move to another place. So a large part of the work in the field is not to do the experiment itself, is to hike through the forest to find the next trail. Once you find it, you can do the experiments multiple times, but it's not enough to be able to look at. Like we can do it 2 or 3 times, but it's not like you're going to have be able to test for memory effect. And again, also, that obstacle we put along the way is just one of the thousands of obstacles they have to tackle along their single trail, meaning that even if you were to repeat the experiment here, the learning is distributed over the entire trail, meaning that your effect here might be completely washed out in the mix. So it would be very complicated to test that. This being said, there are, in other situations, examples of colonies that can learn collectively certain features. So it's technically possible, it's just practically impossible to do in the field. That's the end of my talk, by the way. So.
Wonderful.